Hi, everyone, and welcome to another We Talk Science podcast. Today, I have some very special guests on the show. The TU Delft iGEM Team Focus. These guys work in TU Delft in the Netherlands, and they are basically looking at a project that looks at the locust crisis. So, you guys, please introduce yourselves.
Hi, thanks Sheba for having us. I am Gabriella van Leersum and I am this team's outreach manager. So I have contact with you and other people who might be interested in talking a little bit about our solution to the locust crisis.
Hello, my name is Willem van Holte and I am the human practice manager of this team, which entails I look at the social aspect of the problem. So not only at the science, but also the other things that play an important role in solving this problem.
Hello, I'm Iris Forking. I'm the science manager of this year of the Focus team. And my goal is to integrate the wet lab, so the experience we do in the lab together with the models we do in the dry lab, to integrate them and to make a good project out of it.
Wonderful. So let me start off with the project itself. So can one of you describe to me what exactly is the goal of what you're working on? How are you looking at this locust problem? Because obviously in the Netherlands, we don't really have a locust problem, do we?
Yeah, we do not have a locust problem in the Netherlands at the moment, I must say. But it was starting to gain a little bit of traction in the news when we were deciding on a topic. And it became more and more abundant in the news. And it actually that way sparked our interest to look at it.
Okay, so tell me a little bit more about the science then. So what exactly are you looking at? What is the problem essentially?
The problem essentially is that there are locusts, which is actually a type of grasshopper, or multiple different types of grasshopper, which have a very special talent in which they are able to change phase. So they have a solitary phase in which they just live on their own in nature, doing their things. This is like the normal state, you could call it. But then there's also another phase, which is called the gregarious phase. And this actually happens when a lot of locusts are at the same location. And there are actually 3 different triggers for this. So the first is they have little sensory hairs on their legs that if they touch, create all types of different changes within the animal. But this is also stimulated by sight and smell. And this actually changes the locust, so it actually grows in size. It gains quite an enormous appetite, and it starts to reproduce more. And here's actually where the problem comes from, is that because they eat so much, or they're that hungry, and there are that many of them, they are able to devour a field of crop in no time. Quite an astonishing rate, because one single locust is actually able to eat its weight a day, which is only 2 grams. This might not sound that much for a single locust, but actually a desert locust swarm can be up to 12,000 square kilometers, where there are 40 to 18 million locusts in a swarm, or in less than a square kilometer of the swarm. And with this, they actually eat 1 square kilometer of the swarm is able to eat the same food in a day as 35,000 people.
Wow, that's pretty amazing. So 35,000 people. So where in the world is this a problem? The biggest hit areas of these locusts?
Currently, the biggest hit locations in the world is Eastern Africa and the Horn of Africa, south of the Arabian Peninsula, and the Indian-Pakistan area.
And are we how much of loss are we talking about then with these when they swarm and these locusts actually swarm?
Okay, yeah. So the World Bank made an estimate in March based on the projections of the Food and Agriculture Organization, and they calculated that in East Africa and Yemen alone, the damages and losses could amount up to as much as 8.5 billion US dollars.
Okay, so that's quite a huge loss that we're talking about. What is the way that, how did you guys tackle this problem?
This is Iris. So we want to tackle this problem, and you might think, why is this problem still there? So we also thought this. So we went looking around on the internet, and we looked at how they tackle the locust crisis now. And they use chemical pesticides. And a big problem with the chemical pesticides is that you need a huge amount because these swarms are very big. But a very big drawback is that it's not specific, because the bees are also targeted and killed. And even for humans, these toxins are harmful. And a different alternative that they're currently also using is a fungus, which is called metarhizium. So this is better because it's more specific to the locust. But the problem is that this is too slow because it takes around 7 to 11 days. And in this time period, these swarms already increase of size. So we looked at all these things that we're doing now, and then we came with a list of 3 things we wanted for our biopesticide. We wanted it to comply to. And the first thing was that we wanted it to be specific. So this means that, for example, bees shouldn't be harmed or other insects. And the second thing is that we want human safety. So it shouldn't harm humans. And if we spray it on the crops, we still want the crops to be edible. And the third thing is that we want it to kill fast. So unlike the metarhizium, which they're currently using, we want it to work faster. So then we're looking around and we thought, let's see if we can produce a toxin. But the problem with toxins is that they're not very stable because of the UV light and the heat. So then the idea was, what if we do a Trojan horse method? So this means that we produce the toxin inside the locust guts. So then the idea of one of our teammates was that we use a bacteriophage, but that's a small virus which targets a specific type of bacteria. A bacteriophage injects its DNA into the bacteria, and then it uses the bacteria as a factory. At this point, it hijacks the machinery. So this means that the bacteria only produces the injected phage particles. So we're going to use this method to produce a toxin inside the locust gut, and then hopefully from inside out kill the locust.
That's a really nice strategy, I have to say. So tell me where does the dry lab and the wet lab part come in here?
For the wet lab, because maybe this is also an important thing to say, is that our project, I think it started in March, and we're working full-time until October. So we're not going to finish the whole project and really produce the biopesticide. So what we're doing now is we're trying to do a proof of concept. So what we're first doing is trying to show that we can engineer the phage. So for a simplified version, we're going to insert GFP inside a T7 phage. The T7 phage is a phage specific to E. coli, because this is a bacteria which is easy to use and there are no restrictions. So we're going to replace a non-essential gene inside the phage with GFP and then infect E. coli. And then we hope to see that E. coli produces GFP. So with this, we really want to prove that we can engineer the phage. And then later on, we'll try to produce the cry toxin, which is a toxin against insecticides. And we found one which is specific to another type of grasshopper or to another type of locust. And we're going to produce this and see if we can show with experiments that it's produced. And we also have some modeling, and Willem can explain that.
So for the modeling, even though we have access to quite good facilities and resources, it is still very difficult to transport laboratory results into the real world. But luckily for us, computer modeling can help us out with that. And this is what we call the dry lab. And with these computer models, we actually are trying to simulate the bacteria and phages in the population together to investigate how many phages or phage infections rather we would need to create the right amount of toxin to actually kill a locust. And we are actually also working on a second computer model in which we focus entirely on one single bacterium. And we try to understand the different trade-offs that are made within the cell when it is infected by bacteriophage, to gain a better understanding of how much toxin one single cell is able to produce.
What are the kind of scale models are you looking at to make sure that this is scalable in the real world?
Currently, what we're working on the scale models is still on the size of the locust itself or its gut, to actually know what things we need to adjust to make it workable on the single population. We have also looked a little bit into population dynamics, because we know for locusts they reproduce 20-fold each generation, which means that if we want to maintain a stable population of locusts throughout the world, we need to kill somewhere between 90 and 95%.
Okay, so that was my next question. If these locusts obviously are reproducing really fast. If you were to infect one locust with this bacteriophage, would it be transmitted to its offspring, or would you have to keep reinfecting them?
This is Iris. You have to keep reinfecting the locust because they're only transferred from bacteria. The phage infects the bacteria, then the bacteria lyses, so it sort of explodes, and then the phages are spread. But when it would get offspring, the bacteria are not transferred from locust to the locust nymph. I think the correct name is nymph. So this would mean we would have to reinfect the nymphs.
Okay, so again, if the locusts are replicating really fast, you would basically, you're thinking about, several waves of sort of transmitting the bacteriophage into these locusts, hopefully killing, what did you say, 95% of the population, correct?
Yes, that's correct. And we also want to see how fast we can kill the locust. So I think in the models, we're also trying to figure out how much toxin we can produce and then see if we can have an estimation of how fast it is. Because our goal is to do it as fast as possible. So hopefully in the time being, no new locusts are born.
Right. Okay. So it's like a race against the clock, really.
Yes, definitely.
Okay. Can you talk a little bit about those results, maybe? I mean, do you have any data yet to know how many times, let's say, you would have to infect the bacteria, the locust with the bacteriophage, and how efficient would it be? Do you have any data about that you can talk about?
Currently, we do not have exact data yet. I guess we are still working on this. So we're performing a linear stability analysis on our models to better understand where the stable points lie, so we can actually see to which extent it can spread or might not spread.
Okay, that's interesting. Okay, so let's look at the social aspect of this. Obviously, this technology would affect and benefit people in the countries that you've mentioned, like Africa and India and Pakistan. Are you collaborating with anybody on the ground in these countries?
Yes, certainly. I think it's very important for us to actually collaborate with these people because things work differently here in the Netherlands than it might be in Africa, in India, Pakistan. So this is very important to us, and we've already had some interviews with different kinds of people. So we have spoken to the CRC, which is a commission of the Food and Agriculture Organization that is responsible for the locust control central region, and this is mostly the Arabian Peninsula and the northeastern part of Africa. But apart from that, we are also going to speak at a locust conference on August 10th. And from this conference, we hopefully will get some connections on the ground, actually, because we found to actually acquire these connections can be quite difficult, especially now during the pandemic.
Okay, so what would this look like? How do you guys envision this pesticide looking like? I mean, is it going to be a spray? Is it going to be what is the delivery method of this bacteriophage?
So this is Iris, and we talked to some experts on how we can deliver our phage or biopesticide, and they do say this can be quite difficult. there have been companies that also did some experiments in the lab for biopesticides, and when they applied it to the field, they saw that stability was different and that locust behavior could also influence this. So for our project, we have some ideas, but of course we still have to see if that would be feasible. For our project, I think the most important thing we have to consider is that our phages have to be inside the locust gut. So this means that the locusts have to eat our phages. So for now, our main idea is to or spray it on the crops or to make a bait which the locusts eat. So then that the bacteriophages enter the locust guts and they can infect the locust gut bacteria.
That's really interesting. So going back to something that you mentioned before, the three things that were important to you when you were looking at this particular way of tackling this problem, you talked about specificity. What are you doing in terms of that? Have you tested other organisms or other, are you going to test, I should probably ask, other sort of insects that might have these bacteria also in their gut that might end up killing them as well, like the bees?
That's a good question. For specificity, we found toxin, which is the cry toxin, and a paper describes it for being very specific to the locust, a different type of locust, however. Mm-hmm. It's the Locusta migratoria, and they found that it was very specific to only this type of locust. So by expressing this toxin, we'll ensure that, for example, bees or humans are not harmed. Mm-hmm. I should, however, say that we ourselves are designing a project for the desert locust because we saw that this desert locust has a bigger economic impact on people. And we did contact these researchers to ask if there was a similarity. And some research said yes, and others said that they don't know, so this should be tested. Mm-hmm. We do know that this toxin is not harmful for other insects such as bees, and also not for humans. In addition, we also think it's very important that it doesn't harm humans. And we ensure this also by using bacteriophages, because for humans, the pH of the gut is around 2 to 3, I think. And for locusts, it's around 6 to 7. And bacteriophages only survive a pH of 6 to 7. So this means even if humans would, for example, ingest it, the bacteriophages would not survive and wouldn't be able to infect our gut bacteria and produce the toxin in our bodies.
Okay. Please tell me a little bit more about the human safety part, because I'm guessing that if there is a pesticide of any kind that comes out into the environment, there has to be a lot of rigorous testing to make sure that there is no effect on the humans. So what is the normal procedure of this? When you're actually testing a pesticide in the field?
Willem here. There are, of course, all types of different impacts, like the specificity. So I think, first of all, the primary thing you have to show when you develop such a pesticide is, especially for biopesticides, I must say, you must prove that it only infects insects and not warm-blooded animals. This is like the primary part where it all starts. If you cannot prove this, then it will be very difficult to actually gain acceptance of your pesticide. Another part that is rather focused on human safety is that even though you might have information saying that it does not affect humans, still the infection temperature should not be the human body temperature. So this shouldn't be 37 degrees. So this was almost the case for the Metahesium biopesticide, which has a peak at 35 degrees. So this was just about cleared for human safety.
Okay, interesting. All right, so tell me a little bit more, just to change gears a little bit, about this particular iGEM project that people or students are actually involved with. Can you just tell me a little bit more about that? Is it kind of a competition, if I understood correctly?
Yeah, let me tell you about it. This time it's Gabby speaking. iGEM actually stands for International Genetically Engineered Machine, and it's a non-profit organization that focuses on the advancements of synthetic biology and the education thereof. And they organize a yearly competition. So that's the iGEM competition that we're talking about and that we are taking part of. And about 350 teams participate each year on different levels. It's either school teams or undergrad or overgrad. And so that's roughly 6,000 people per year who actually sort of will try to come up with new solutions to real life challenges on a global and regional scale. So it's quite a large competition and quite a large contribution to the field.
Cool. So you guys are putting your hats into the ring, and hopefully your project will gain some recognition there then.
Definitely. Not only the project, but also the science, obviously. And also something that is noteworthy is that iGEM doesn't only care about synthetic biology, but also really about the integration of advice from experts and really the outreach and science communication and things like that. It's very educating the scientists of the future how science should be done, and not only focusing on the technology, but also on everything that goes around.
That's pretty fantastic. I mean, that sounds like a really, really good idea, a really good way to move forward, especially to get young people interested in science, of course. Okay, so let me ask you one question. Let's say if, I mean, obviously there'll be a lot of projects that are being presented at this iGEM thing. What is the future of this one? Because, I mean, obviously it is solving a very, very pertinent world situation problem. So let's say, when you, after October, when you have your results and you present your work and all of this, is there, someone who's going to take it forward again? I mean, because this sounds like a very, very interesting, almost like a whole, probably a PhD project or something like that. So what is the future of this particular idea?
Well, honestly speaking, it's not something that we've talked about as a team, but we do have a little bit of a entrepreneurship going on to see how we could actually bring this product in the market if we were to get that far. Of course, we're all, the competition is towards the end of October, so afterwards we will probably continue with studying and everything that we still have to do with our lives. But it is something that could be picked up later on by either companies or the FAO that also really works with this locust crisis, because we are not only setting up the scientific basis, but also a little bit of the entrepreneurship part of it.
Thank you so much. I mean, this was so informative, and you guys obviously have the tools as well as the enthusiasm to take this forward. And I wish you all the best. And I hope that, you get recognition for this really, really important work that you guys are doing. Are there any last words that, that you want to say? Gabriella, any last words from you?
Well, you're really putting me on the spot here. We'd really like to, get a little bit of awareness on the locust crisis. That's also something that we really want to get out there. Obviously, it's very much on the news lately, but it doesn't get as much awareness as the amount of damage that's being created. And thank you very much for having us. That's also something that I'd really like to add.
Willem, do you have anything, any last words that you would like to add to that?
I would like to add that a little bit, maybe more locust awareness, and just a little number I want to put out is that the FAO predicts that this locust crisis affects up to 10% of the world population. And I think people do not recognize this as such a big problem. So not only in economic damages, but we're also speaking about food security. So this is like a primary human need to eat. And it's a pity that there's so little attention, I feel, for such a big problem. Great.
Iris, any last words from you?
Well, I think I agree with Gabs and Willem, and I think we're trying our best to make this project as good as it is, and even if we can add a little bit to helping this solve this crisis, that would be amazing.
Well, I just wanted to say thank you again to all you guys, because this has been really, really interesting for us, and I'm definitely sure it'll be very interesting for our readers as well. Thank you again, and good luck, and I'm sure that we'll hear the results of the competition probably at the end of the year, I'm guessing.
Yes.
Okay.
Thank you very much. Thank you.
Thank you.